We're really excited that you joined us again this year for LABEST. Now I'm really excited to introduce our next panel, moderated by Paul Webb from CIRM, intitulated Challenges and Prospects for Cellular Cancer Therapies. Welcome them on stage. Thank you.
So welcome, everybody. I'll just introduce myself very briefly. I'm Paul Webb, and I work at CIRM. And I think, as you all probably know, CIRM delights in giving awards to enable spectacular clinicians and scientists to advance therapeutic development. Often forgotten is that we're also very interested in making sure that those therapeutics are readily accessible to the people of the state of California. So what we have today is it's absolutely spectacular panel that can address both of those aspects. And what we're going to be discussing are cellular therapeutics for cancer. So I'd like the panel to introduce themselves. Tony?
Hi, I'm Antoni Ribas. I'm a professor of medicine at UCLA, and I've benefited from working with CIRM and being funded by CIRM, so thank you.
Hello, my name is Alice Pomponio. I'm the Senior Vice President for Corporate Development and Strategic Partnerships at the Parker Institute for Cancer Immunotherapy. It's a mouthful. And I'm sitting right next to our PICI director here at UCLA, so very proud to support the activities on site.
Kevin Cognetti, I lead strategy at Kite, and we're one of the leaders in CAR T therapy.
Hi, everybody. I'm Zach Roberts. I'm the head of R&D and chief medical officer at Allogene Therapeutics.
Okay, so I'm going to begin. So as a slight bit of background, I think there's 7 approved CAR T right now. Is that correct? But I can definitely count them on my fingers at the moment. Those are for hematologic malignancy. And what I'd like you to address is where we are with solid tumors. Where do you see the points at which solid tumor development is slowing down, and what are the opportunities that are coming through? So Tony, can I begin with you?
Well, I think it's targets.
Yep.
The first thing is, if you're going to redirect the immune system to cancer, you have to be pretty sure that the cancer target is not on the normal cells. And in solid tumors, it's been difficult to do that. In B-cell leukemias and lymphomas, we learned by doing CD19 and 20 and 22 targeting that we can live without B-cells for a period of time while we treat the cancer. But with other for epithelial cancers, we cannot live without epithelial cells. So that, I think, is the problem number one. The next problem is that once you get T cells into a solid tumor, the solid tumor is a place where the pH, the metabolites, everything is wrong for a T cell to proliferate. So that has also limited how cell therapies work.
Okay, so I'm going to go slightly out of order because, Zach, I know you'd expressed a strong interest in commenting on this question.
Yeah, so we have a solid tumor program at Allogene. It's been open for several years. It's targeting CD70, and I couldn't agree more with Dr. Ribas' point about finding the right targets for cellular therapies. CD70 happens to be a pretty good one because it is really isolated to the tumor in most cases. It is slightly expressed in some other cell types like immune cells. And as an allogeneic cell therapy company like Allogene, we actually leverage that expression on the host immune cells to our advantage. So as we're targeting the CD70 with our CAR T cells, not only do we attack the CD70 expressing kidney cancer, but we also attack the cells in the patient's body that are trying to reject the CAR T cells. And that actually stimulates the CAR T cells to divide and expand. In our phase 1 trial, we have seen 31% of our patients with metastatic RCC achieve a very deep and quite durable remission. We've got patients now out past 18 months with only a single dose of allogeneic cell therapy. So I agree that there are, the hill is quite a bit steeper in solid tumors. Target heterogeneity is another issue. Sometimes only a subset of the cells in a given tumor express the target of interest. But I think that there are some bright spots, and I think our phase 1 experience using our Allo316 product in RCC is a great example of what can be achieved with cell therapy in solid tumors.
Well, thank you. Can I turn to Kevin?
Yeah, if I could add, congratulations first off, because the durability is another area where solid tumors, because of the heterogeneity, hasn't shown as much promise. And what we're seeing is that if you can add armoring or an enhancement, you may be able to pull a longer durability and persistence of the CAR T cells. So at Kite, we are entering into glioblastoma and hepatocellular cancers with that type of focus. Now, when you continue to add pieces to the puzzle, the payload becomes an additional problem that you have to solve through. So we're working hard at that, but I think the promise of solid tumors is coming now, and it's coming forward quickly.
Alice, would you like to comment?
You know, I'll just kind of give more of an ecosystem and funding and financing layer and perspective, which is we do have to break the paradigm, right? And so right now, CAR T is perceived to really only be for blood-based cancers. And so all of the science that's just been described absolutely needs to be addressed. But then there's innovation even in the delivery, right? Because these are hard places to get to in the body. And the tumor microenvironment and the heterogeneity is also something that can be tackled both medically as well as physically. But I do think that this space, while it has great promise, has been underinvested. And we do need to think of how to make the R&D pathway much more efficient and effective. And so that is one area where PICI has been able to bring together the highest concentration of IO experts in ways that perhaps would not be normal or natural because of our ecosystem and the ability to collaborate with new sources of funding and capabilities.
Okay, and are there particular solid tumors that would lend themselves to CAR T therapy, in your opinion, or not? You know, they open.
What is Kevin?
Go ahead. Well, we think ones that are more concentrated, the glioblastoma, you can target it directly through the Humaya. Hepatocellular sits in the liver pretty stably, and so you can target it differently. But Zach has seen great results with RCC as well.
Yeah, and I'll say that there's been some interesting early data using Claudin 18.2 as a target for GI malignancies. We've seen proof of concept, not as prominent with cell therapies, but with T-cell engagers against DLL3, which is a target that's expressed in small cell lung cancer. There's no reason to think that a CAR T-cell couldn't mimic or even beat the signal that's seen with a T-cell engager in that context. So I think that there definitely are some targets that are worth pursuing, and I would agree with GBM it also expresses CD70. So there are some bright spots for sure, and I agree with Alice that more focus and more dollars are required to generate that proof of concept to break through that hematologic barrier.
Yeah, to add to that, the investment dollars, we believe one of the big reasons why we're not seeing as much investment is we haven't broken the paradigm in blood cancers. We're still only getting to 2 out of 10 patients. The ability for us to change that paradigm and show that CAR T is not a niche product, that CAR T can cure on the masses, is going to be a huge importance to people flowing investments further and further into solid tumors and beyond.
Right, well, good. So I'd like to switch topics to the next topic now, if I may. And it's a very kind of open-ended request to you all. It's basically to ask if you'd comment on safety, durability, and efficacy of CAR T therapies. And just tell us your opinions, where we are on those issues, and what you think the key challenges are and how we're going to overcome them. And in this question, we're not restricted to solid tumors. I think you're already anticipating some of this, Kevin. So if I may, can I begin with Alice this time?
Oh boy, I'm going to cede the floor to others so I can add to it. But I will say that, these broader systemic questions, especially that, at the broadest level can only be divided at a patient population level, are, again, very difficult to pursue outside of an academic or government-funded environment. And so companies are not incentivized to do so. PICI, Parker Institute, did previously invest significantly in developing clinical trial cohorts, particularly Radiohead, which does look specifically at adverse events related to, immunotherapies. And so I think that is almost an infrastructure play, if you will. It's a public good that now researchers can lean on, but it's really taking it to the next level and asking those more targeted questions about what to do with the knowledge.
So, Tony?
So it's target specific. So we're going back to the same things where if you target something in the cancer and it's expressed in normal cells, then there's the safety issues. There's some targets that are expressed at different levels between cancer cells and normal cells, and then you can have a therapeutic window. If we go back to the CD19 CAR when Kite started, and there were some academic groups doing CD19 CARs, we didn't know about the neurotoxicity. We didn't have a word to describe the cytokine storm that we described afterwards. And all of those were things that the people learned by doing it. But if the key thing is if the therapy works, we're very good at adapting to understand what you do to give it safely. The good thing about CAR Ts is that it's a one-time deal. And if it works long-term.
So I think that toxicity is certainly one of the main drivers of the restricted access to CAR T cells. And I would agree with Tony that it is sometimes target dependent, but also is sometimes target independent. And even the cytokine release syndrome that is often following CAR T cell infusion is more of a non-specific toxicity than it is a target-specific one. It is indirectly related to the abundance of target, but the absence of B cells is not what's driving the cytokine release syndrome. So the toxicity management still today with autologous CAR T often requires that these patients be near the hospital for a certain number of time. Often, maybe 50% of the time or more, they require a brief in-hospital stay to manage these toxicities. So toxicity is certainly one of those things that is contributing to the restricted access. The other part is the infrastructure that's required to the endeavor overall, what an institution needs to invest in to be able to collect the cells that are required for manufacturing of autologous products, to administer the product safely. There is a barrier to entry that is pretty substantial, and there's accreditations like FACT that comes with all of that infrastructure that sort of signs off on your ability to safely administer autologous products with chain of custody and chain of identity and so forth. At Allogene, our solution to both of these problems is through, number one, an off-the-shelf product that is centralized manufacturing, so there's no need for leukapheresis, and then through trial design. So we are running now our pivotal program in frontline consolidation of diffuse large B-cell lymphoma, in which patients have very minimal disease at the time that they're treated with their CAR T cells. We find patients who have completed frontline treatment, who are in a remission, but are still have detectable molecular disease in their body through a test called MRD. And those patients who are MRD positive are very certain to relapse, usually within 4 to 6 months. And if we administer CAR T cells to those patients, we can actually do that without any of the infrastructure, without factor accreditation, and we have seen now in our early data cut, no CRS, no ICANS, no other toxicities that would require a patient to be near a hospital or in fact even need to be admitted for toxicity management. So if we can successfully run this study and commercialize our product in this context, we believe that actually we'll be able to totally break that access barrier, democratize CAR T, and be able to administer it in community oncology practices that currently are not offering autologous products.
And just building on what Zach is saying, I think there are better management of bridging therapies and things like that to reduce the tumor burden in order to reduce the safety and adverse events. But also, applying different science to it and developing CAR Ts in which many of the companies now are taking dual targeting, different signal domains, and things like that that will drive a different safety profile overall, which then you don't need all the treatment beforehand, all that chemo, loss of hair, et cetera, you can move right to the CAR T and still get that promise of the one and done treatment. So there's two ways to do it. And one way, I agree, is to prep the patient differently. But the other way is to figure out the science and make sure that you bring better medicines to the market.
Sure. And following up on that, one of the observations from CAR T trials has been the differences in durability of response, ranging from people who have extremely durable responses to people who see a terrific response which goes away quickly. What's your opinion on the main drivers of that phenomenon?
It's almost certainly multifactorial. There are some cases where we can clearly find a smoking gun as to why somebody doesn't respond or has a short-lived response. Coming back to target expression, that's one that comes to mind. If you lose, in the case of CD19 malignancies, if you lose that target on the surface of your tumor cells, they become invisible to the CAR T, that tumor's going to come back. Sometimes the product itself does not pack the punch it needs to pack. Sometimes the patient, the T cells are not super healthy, and so if you're making a product from that patient's T cells, you're sort of doomed from the start, and often you don't know that until you've infused the cells and you don't get the response that you desire. Sometimes the tumor is too bulky, and it overwhelms the ability of the CAR T cells to expand and eradicate that disease. And there's probably dozens of other explanations that we're only beginning to understand.
Sure, and to what extent is this a technology question? Do you think can it be improved within the CAR T? Immediate steps to achieve this, or?
Yeah, having the path to be able to treat solid cancers will require having armored CAR Ts. The target is important, but then how the T cells that are going in are acting and maintaining function. We know that every time you turn on an antigen receptor on a T cell, the T cell is trying itself to turn off. So whenever you, in some cancers, the CARs can signal well because they have signal 1 and signal 2. But in other approaches like T cell receptors, we're seeing that mostly are transient responses where the T cells function up to a point where the T cell gets exhausted. So if you have that T cell then armored with something that overcomes exhaustion, then we could have durable responses.
Great, thank you. So, go ahead, please.
Yeah, I think there's two different things to bring up here. One is, yes, technologically, we can have less antigen loss. We can go and target multiple tumors. We can add to the science. But there's also a technology around patient identification. Mm-hmm. Not all patients are the same. And even though they're categorized as a large B-cell lymphoma patient or a mantle cell lymphoma patient, there's subsets within those that may have different characteristics. And learning those and getting more predictive, working with people like the Parker Institute to understand that is an important next step as well in the durability question.
All right. So if I may, I'll switch topics, and this will be our last topic for today. And this is an odd one because it's patient access and infrastructure. And it's really not a separate question from the first two topics when we come down to it. And I don't want to give anybody the impression that it is. But it's been a challenging area for the whole field for quite some time. And the question I've got is, what do you think it would take to scale these therapies beyond top academic medical centers? And who should be at the table for those discussions to move it beyond the top academic centers? So I'm going to begin this time with Kevin, if that's okay.
Well, I think we need a cross-industry, across trade association, technology groups to solve this problem. And There's a work that's being done through the CAR T Vision work, led by different medical professionals and different industry partners, that is looking at this very problem, and it's multifactorial. There's a reimbursement friction that we need to solve around these more precision medicines that are more expensive. There's a second around access, as you said, fact accreditation that Zach brought up earlier, and how do you accredit different sites beyond the academic centers, but make sure they still have the competencies and the quality in order to get CAR T treatment closer to the patient, because studies have shown that the closer you are to your community and to your family, the better outcomes you have. And so that's the type of work. Individually, Kite is working on opening up more and more centers, but I think it's a cross-industry and cross-community practice endeavor to really change the paradigm of how this works. Because it's not just CAR T, it's going to be more and more gene therapies and other high-priced medicines that our system isn't built for.
Zach?
Yeah, I mean, I would agree. I think in order for us to truly achieve what we sought out to achieve when we designed this field 10, 15 years ago as a group, we need a paradigm shift. We can't really continue to iterate on the existing paradigm and hope to incrementally get our way to 80, 90, 100% of patients who would be eligible for a CAR T to actually receive that CAR T. I don't think we're ever going to get there. We've been at this for 10 years in the commercial context, and we still are only treating about 15 to 20% of patients who could benefit from a CAR T or actually receiving CAR T. Think about that. I mean, that's the overwhelming majority of patients are not going to enroll in a clinical trial that leads to New England Journal publication, right? These are folks who want to stay with their home oncologist who took care of them when they were first diagnosed. Even patients who could go across town 20 miles to a CAR T center often won't do it because they want to stay where they're comfortable. And so the paradigm shift that we at Allogene see is to try to break free from this, what is required and has historically been required to administer CAR T. And you can't do that within an autologous context. You just can't. You either have to go to an allogeneic, as we've done, or you have to do something new and novel, which many or others are looking at in vivo products that could potentially shift the paradigm. One of the things that we are excited about at Allogene is that we have an ongoing pivotal study right now. I mean, we are doing something. Half of our sites in our pivotal trial right now are community practices. A substantial subset of those, are sites that have never given CAR T cells to anybody before. A third of the patients in our recent interim data analysis who received cema-cel at sites that had not used CAR T, at least not routinely. And they're doing this as outpatients. The patients are coming in, they're receiving their cema-cel infusion in the infusion clinic, and they're going home, and they're never coming back into the hospital for toxicity management. They just then resume normal care. If that is something that becomes real in a FDA-approved commercial context, that will break the paradigm that we need to break. And so I think collectively as a field, we really need to think outside of the box that we created 10 years ago with these autologous products and find out how we can get to 80% of the patients instead of 20%.
Right, so Tony?
We're in a time where it's really exciting with the new technologies that are being developed. The in vivo gene editing is something that it's going to revolutionize what we're doing with CAR T therapies and redirecting T cells in vivo. That's closer to a drug than it is to a cell therapy. It's not a cell therapy anymore. But for certain targets, the autologous or the allogeneic cell therapy platform is a validated platform. So I think we cannot shift everything to the in vivo gene editing because there's some targets that don't want to have too many questions every time you do something. So a validated cellular platform may be advantageous. We have Gay Crooks here in the room. Gay is developing from a stem cell into a T cell and being able to gene edit the cells in between. So we have a lot of science that's turning a reality that it's allowing to address the shortcomings that we've had so far with the cellular therapies for cancer.
So, Alice?
And I would just add, again, we're in a unique moment in where we are with our market and our industry. And so we can't afford to rely on discovery-led approaches to be the sole way that we change these paradigms. So on the Parker side, when we think of, what can we, from a financial standpoint, back that will be disruptive in 5 to 10 years, we are absolutely looking at the in vivo CAR T. We're looking at even cancer vaccines from a durability and addressing treatment as well as prevention, but also thinking of how to generate more efficiency in the way that we conduct R&D in this space. And so absolutely what Dr. Ribas had mentioned, but bringing together in a pre-competitive fashion multiple stakeholders who have underlying questions that they themselves cannot justify the expense, but could potentially pool those resources working with a network like the PICI network to answer fundamental questions that could unlock the field will be how we revolutionize. So moving more towards demand-led and actually embedding that into the product development design criteria and then building the confidence in an alternative pathway for product development is how I see addressing these bigger systemic level challenges.
Excellent, thank you. Is there any world where autologous ex vivo manufacturing could be taken out to community centres?
We're doing it.
Yeah.
So we.
Before.
Okay.
In the future, will this persist? Is really what I'm trying to say.
Will this persist? I think so. I think that as the treatments get safer, as the medicines get delivered in a reliable fashion, as the community gets more comfortable with the management of it, whether an allogeneic product or an ex vivo autologous product, that will create the footprint for them to continue to do it. And so there are structural barriers with the reimbursement when you get reimbursed, reliability on the reimbursement. There's also structural barriers in setting it up from a 24/7 nursing care and things like that. But those are overcomable if the finance works at the community oncologist center. If they can make money on it, they will continue to do it. And it's our job as a community to make sure that that is pulled in the right way. Now, will it go to every mom and pop community center? Probably not. This is still an involved treatment. It still requires a lot of investment and a lot of infrastructure, but we believe that it will get to the major centers in which it'll make it much closer to home and much more patients servable.
Well, I wish you could all talk more, but we're actually getting down to about 2 minutes. And what I'd like you to do, one by one, is just give a final take-home message to the audience of, perhaps a single bullet point of what you think the most important developments in the near future are and ought to be. So can I begin with Tony?
We have to develop curative therapies for cancer, and we have to try more things in humans or bring more science to patients. The funding agencies, PICI, CIRM, the regulatory agencies, we're seeing the FDA changing the paradigms and saying for early clinical trials with cellular therapies, we're not going to require everything that we had required before because that limits innovation. We're seeing what's happening in China, where there's the hospital-based approaches are being able to test multiple things, iterate fast so that you can have active therapies in patients earlier. The problem is there, and we have the best tools ever now and the best science. So I think there's a bright future.
Alice?
Yeah, I would just argue that in addition to scientific validation, we should be thinking of sustainability and scale. That is particularly important for this modality because even what was mentioned this morning with Lily, thinking of outside the U.S., very different healthcare archetypes, very different populations. And so embedding those questions early on such that the science can address those and we don't have to do it through health system, policy, reimbursement mechanisms to drive the outcomes that we hope to achieve.
Right. Kevin?
Yeah, I'll just add, because I agree fully with Alice, that it's going to take a village. And therefore, we have to work together in a pre-competitive way in order to make this happen. It's not going to be one group that changes the paradigm. And if we all work together, we can bring these medicines to more and more people in a more effective and more cost-effective way.
Right, and I'll give the last word to Zach. Okay.
Sure. So I'll echo the access and scale because I think that's critical that we solve that problem. Again, I come back to this 15% of patients who could get a CAR T actually receive it. That to me is almost shameful as a field of what we've been able to achieve. Stepping beyond that access and scale, I think patient selection, finding the patients who could benefit and treating them as early as possible is going to be a big part of the story in the next 5 to 10 years. And that, to me, I think is going to open an entirely new way of thinking about how to develop these novel agents, not just going into patients who have got first, second, third line failures, but actually finding patients early, maybe at the time of diagnosis, maybe shortly thereafter, and optimizing the clinical trial design to improve both safety and efficacy outcomes. I think there's a lot that we can benefit from that strategy.
Great. Well, I'd like to thank you all. What a great set of remarks and what a great panel. And maybe we can applaud everybody.